Hello, I'm pleased to share our contribution to the study of wastewater surveillance for SARS-CoV-2. We represent the Molecular Monitoring Lab at Grand Valley State University in Grand Rapids, Michigan. I'm Dan Adrian, and as you can see, I'm in the Department of Statistics, so I'm not a biologist by training. So though I've been coached well, I'll do my best to speak accurately about these biological issues. Our poster is primarily a collection of visualizations numbered 1 through 7, so I'll present some comments on each of these in order. Thanks to a grant from the Michigan Department of Health and Human Services, we've been collecting biweekly samples from wastewater treatment plants in the Grand Rapids area since June 2021. Here we focus on plants serving Grand Rapids itself, along with 2 suburbs, Granville and Wyoming. The map here shows the zip codes included in the sewer shed for each plant. We used these zip codes to connect the population above ground to the subterranean wastewater. We collected the daily COVID-19 cases by zip code reported by the Kent County Health Department. We ran each wastewater sample through our ddPCR to measure the number of SARS-CoV-2 copies per 100 milliliters. An initial question was whether we could reasonably combine the samples from different wastewater sites. This plot shows that the answer is yes, because the viral counts from different sites show similar trends in time. As you can see, the lowest, smoother lines, the thicker lines shown here, are close to each other. You may note that this plot shows 2 additional sites not identified on the previous zip code map. These sites are the intakes at Byron Center and Kentwood. Downstream, these 2 sewer lines both entered the Wyoming plant. We use different sampling methods at the 2 intake sites than at the 3 plant sites. The intakes, we use grab sampling, where the wastewater sample is all collected at once. In contrast, the 3 plants use composite sampling, where the sample is collected at multiple times over a 24-hour period. Given that composite sampling better accounts for variations in the wastewater at different times, it's not surprising that the grab sample sites showed more variation, as can be observed in the having more spikes on the previous plot, deviating from the smoother lines for the Byron Center and Kentwood, the red and blue lines. And these larger deviations can also be seen by the larger spread of the box plots for the Byron Center and Kentwood sites on this plot. We aggregated the COVID-19 case counts and wastewater data across sites and to the weekly level. This figure shows both of these time series on the same plot to emphasize the strong correlation between the wastewater and clinical data. Both time series increased through the Delta variant period until peaking at the start of the Omicron variant period. Then both time series fell together before rebounding together. However, around June or July 2022, the public health data became less reliable, and cases likely undercounted due to the increased availability of at-home testing and decreased reporting to public health agencies. After this point, we see a breakdown of the correlations observed earlier. The reported cases fall in this period, while the wastewater data is relatively steady. This plot provides another way to visualize the relationship between the clinical and wastewater time series, where the scatter plots show the progression of the data in time using the arrows. It shows that the linear relationship was strong during the Omicron and Delta variant periods, but weak in the last Omicron subvariant period. Also, the long run of points that are above or below the straight line fit demonstrate time dependence in the data. In other words, weeks with higher than expected case counts, according to the relationship with the wastewater, tend to be followed by other weeks with higher than expected case counts. And the same is true for lower weeks tending to be followed by lower weeks. If the data were independent in time, the case counts should cross the fit line at least once every few weeks. A hoped-for application of wastewater-based surveillance is the prediction of future trends in disease transmission for public health preparedness. This application requires correlation of past wastewater data and present clinical data. In other words, correlation where the wastewater data is shifted back. This plot shows the correlations when such shifts are applied. While the strongest correlation occurs for the unshifted time series, that's with the 0 week shift observed on this plot, we can see there are moderately strong correlations for the shifts of 1 or 2 weeks in either direction, suggesting that there is some forecasting ability. The strong correlations and linear relationships shown previously lead to a simple linear regression model fitting the actual case counts well. The predictions produced by this regression model, assuming time independence, are shown by the blue line here. However, taking advantage of the time dependence previously pointed out through an AR1 model greatly improves the predictions. This AR1 model takes into account whether the previous week's case count was above or below the model fit and adjusts the current week's fit accordingly. It should be noted, however, that the AR1 model requires current and accurate reported case counts, which, for instance, aren't currently available for COVID-19. To summarize our findings, we note that our data collected at multiple wastewater treatment plants in the Grand Rapids area indicated that viral counts can be aggregated across plants. Also, we found that composite sampling is less variable than grab sampling, but both methods are valuable for modeling infection level from wastewater, particularly because composite sampling is more expensive than grab sampling. We did not observe statistically significant differences by variant in the relationships between case and wastewater data. We found that offsetting case counts to either lead or lag wastewater viral counts did not improve correlations, though moderately strong correlations were still present at small leads and lags. We found that incorporating time dependence through an AR1 model improves the accuracy of predicted modeling of case counts. And finally, we conclude that wastewater testing has been and will remain an important indicator of community infection level in the absence of comprehensive clinical testing.